So it's me again, and so I told you that my favorite topic in the world was quality scores. So surprise, we get to talk about quality scores again. And these are variant quality scores, where last time we were talking about base quality scores, right? We're estimating the confidence in like a particular base. So now we're going to estimate the confidence in a particular mutation call. And the goal here is going to be, we want very accurate probabilities assigned to every mutation so that we can, downstream, we can use that information in sort of a statistically meaningful way. So let me get this thing. Okay, so here's where we are. Surprise, we're at the next thing. So we've made our mutation calls, and you know these callers are great. They do a fantastic job. But sort of our philosophy with the GATK is that the job of the mutation callers is sort of to output every site that could potentially be variant, right? To be incredibly highly sensitive, but not particularly worry so much about being accurate. The idea being that we're in medicine and we don't want to miss things. We don't want to miss these like very rare mutations that might be the cause of disease, right? So in general, researchers are okay with dealing with large stacks of false positives so that they don't miss that one like killer mutation, right? But you have a lot of false positives, right? So in general, these call sets are infested with false positives. It's like humongous amounts of false positives, not because the callers are bad, just because that's our philosophy with the mutation calling. So because of that, we have this process that you run after mutation calling to filter out the false positives. And so that's called variant quality score recalibration, and it's going to be the statistical machine learning approach that we developed. So let's go back a couple years before we wrote this thing. This is like 3 years now. And it's like the Wild West, right? Where you get these stacks of mutations, and people would come in and they'd be like, oh, I'm a hotshot biologist, so I think that real mutations should look like this. And so I'll write this little filter. I'll say like, we talked about strand bias, right? So I'm a real biologist, and I think strand bias is bad. So I say strand bias less than 10 to the minus 9, you're gone. And then I'm like, well, wait a minute. My project was on Illumina, and they have like some whatever they have, and so I'll say, okay, mapping quality less than 10, that's bad, right? So you have all these hard filters that you like wrote yourself, right? Because you think you have this expertise in the data, and so you hard code this expertise. But then you get to a new project and you have to write a new filter, and every time it's different. And like then some other guy will say he has different expertise and he wants a different filter, and it's crazy. And so the goal of this sort of machine learning approach is to learn these filters from the data itself and sort of not rely on your supposed biological expertise, but instead let the data speak for itself and learn what is the most likely filter based on the data itself. And so this is great for lots of reasons. We don't have to like spend lots of time hand tuning these things anymore. You just run the thing and it always works and it always does the best thing. But the really important thing is that it allows you to trade off sensitivity and specificity now. Where before you had these hard filters, you drew a line and everything that was beyond the line is gone. Where now those things are no longer gone, they're just assigned a probability. And if you want to be very sensitive, you can go down that probability scale. But if you're worried about false positives and you only want to tolerate a 0.1% error rate in your mutation calls, then all you have to do is raise that threshold on the probability. And so it's allowing you to like rank order your variants by how likely they are of being real. So it's pretty powerful. So our goal is going to be to build a model, and just what I said, allow us to rank order variants based on their likelihood. Everyone happy? Okay. So how does this work? Well, like I said, it's a statistical machine learning thing that we'll go through. But basically the idea is that we've developed these like crazy numbers of statistics that we assign to every variant call. And there are things like strand bias, there are things like the read depth or the variant quality score or any number of statistics that people can think of. Anything about the read length or the position in the read or the mapping quality or anything that people want. And what's really fun about this is that people can write their own. If you think you have like some great statistic you want to try out, it's really easy to write your own little Java class, throw it in there, and then you can start using your own annotation, which is pretty cool. So we've had a lot of success in like these huge collaborative projects like the 1,000 Genomes projects, where people will come up with their own things, throw it in and try it out, and like, oh, wow, this is really working. It's really improving the state of the art. It's pretty cool. So we have these statistics, and it's pretty simple when you get down to it. All we did was we just plotted them, to be honest. You take these statistics and you just plot them, and it turns out that real variation tends to cluster together. So like things that are real, they tend to look like each other, and it looks pretty good. And things that were false are like way off in the hinterlands in the distribution. They're like way off the graph. So it's pretty obvious what to do. And also what was pretty cool is that the way we wrote these statistics, they were all Gaussianly distributed, basically. Because they're things like, you know, Gaussianly distributed p-values on different statistical tests, right? So it just happened that these distributions were more or less Gaussianly distributed. So we decided to use, you know, this Gaussian mixture model to like cluster these things and try to figure out what's real. And so the basic principle is going to be things that cluster together are real, and the things that are like around them are probably real. And the further you get away from these distributions, the more likely you are to be false. Make sense? Okay, so let's go through this. So in your VCF file, we went through the VCF file before. You had the chromosome, position, reference allele, alternate allele. But what we didn't say last time was that you have this humongous list of annotations, and this is called the info field. So you have AB, whatever that is, AC, whatever that is. You have just all these things. All these like little letters, and they're equal to numbers. And those numbers are just the value of the statistic. So you have, and they're crazy things. They're like allele balance. We have mapping, RMS mapping quality of all the reads. We have the strand bias score, which we talked about. You have the depth of coverage, the allele frequency in your population, the quality score divided by the depth. Apparently that was an important statistic. There's just tons of these things. And you can add in there whatever you want. And so, like I said, all we did was plot them. And so what I'm going to show you up here is that up here we have this variant call set, this candidate variant call set. It was called on HiSeq with a single sample, our famous NA12878. And on the top, I'm showing you all the variants which were present in this thing called dbSNP. So they're like known variants, previously discovered variants. And on the bottom, they're all the novel variants. So the variants that were not present in this database at the time that we called this thing. So what you see is that I did a Gaussian mixture model where the training data, the positive labels, were the HapMap sites, the ones that were in dbSNP and also present in the HapMap. So the HapMap is this very high quality set of variants. So those are almost certainly all real. It's like a Q35 or something error rate. So those are very good. And what you see is it's beautiful. So those cluster together. You have the heterozygous variants clustering here, and you have the homozygous variants clustering here, and they look roughly clustered, and they're more or less Gaussianly distributed. And so the model that was learned here, it chose that 4 Gaussians was the best description of the data, and that's what it plotted. And then the bars here are like the standard deviation. And the dimensions are, I'm showing you in 2 dimensions, but of course it's like many more dimensions, but I'm just projecting it down to these 2 dimensions. One is variant quality score divided by depth. This is that QD annotation, whatever that was. It's not really important. And the other one is the evidence for strand bias. So as you get at 0 or positive, you have very high evidence for strand bias, and as you get down and down, there's lower evidence for strand bias. And so what you see is that the things look pretty real, and then in yellow, I'm plotting the variants that were in dbSNP, but not in HapMap, and you see a bunch of them are in there. I don't know if you can tell, but there's red and yellow dots here. So a bunch of them are real, and they fall in the clusters, and then there's a bunch of stuff here that falls way outside of the distribution, and they happen to be strand biased. So it's a pretty good bet that those are false. So those are things that are in dbSNP and are likely false. So then okay, that's great. Those are the variants that were already known, but how do we do something with this? So we take all the variants that were novel to this call set and throw them on the same plot, and what you see is a bunch of them fall in the distributions and look pretty good, and you can't quite see it, but there's a bunch of green dots here. The homozygous variants are mostly already discovered because they were easy to discover. So that's why there's not so many here. And there's a bunch that fall off in the hinterlands of the distributions, and those are probably not real. So everyone see how this happens? And so every variant can be assigned a probability based on how, it's just a draw from this distribution, right? It's the probability that you came from this Gaussian mixture. And so, and you can cut at some threshold, and that's the difference between the purple dots and the green dots. So you cut at some probability threshold, and that's why it looks like this sort of strange shape. And then the purple ones would be filtered out of the call set, and the green ones would be kept. What's up?
Sometimes it's clearly biased against mutations. If you use the depth as an indicator of every mutation that has a low frequency, you declare it as false. But it is a countersample.
Yeah. So depth. It's not the depth holding the variant allele. It's the total depth. It's the total sequence depth. So including the reference alleles. So that particular one I don't think is biased.
If a mutation appears in a small fraction in your sample, really small fraction, so that you need a larger depth in order to identify. So obviously you will call it false because you see it only when you have a large depth.
Okay. So there are certainly cases like, for example, exome capture is just one example where depth that's not one of the annotations we use because natural variation in depth is not indicative of real variation or error. But at least I would say in the case of whole genome sequencing for standard human samples, having an excessive high or excessive low depth is an indication of that mutation is actually an error. So what's really beautiful about this is that if you don't like a particular annotation, you just take it out. You just don't use it. Yeah. So anyway, and the other thing to say is that in general, what we're looking for is things that are like way off the distribution. They're like 10 to the minus whatever p-value on the distribution. And so and those are the things that are the errors, not the things that are like sort of a little bit shifted or anything like that. So anyway, we can talk about specific examples. I don't do any cancer sequencing, so I don't know the details. So the other thing I want to show on the right side is that it allows us to give this very beautiful trade-off between sensitivity and specificity. So, and to show that this procedure works with deep sequencing, exome projects, and also low pass projects. So what's happening is that you can decide where to draw this probability cutoff, and you sort of step down and down in the specificity curve. So like you can be very specific and have like basically 100% true rate, or you can get down and down, and these orange bars are trying to represent the proportion of false positives in your call set. So there's like this knob that you can play with to decide where to cut off. Make sense? All right. So I just have one slide with mathematics. So it's a variational Bayes expectation maximization thing. So it's a mixture of Gaussians. So you have this pi mixture, and then the n is a normal distribution. So it's a mixture of Gaussians, and there's just fancy mathematics to do the optimization. So there's a prior on the Dirichlet distribution, which is the Dirichlet distribution, and that's the whole point is to say that it's like a sparse expectation. So like it's trying to trade off having many clusters versus having a small number of clusters. So when you have a small number of clusters, this distribution is large. And so that's how it's able to decide, for example, that 4 Gaussians is the most parsimonious explanation and not like 10 Gaussians or 50 Gaussians or whatever. And then the other thing is just this prior based on the mean and sigma of the actual distribution of the statistics themselves. Anyway, that's just fancy mathematics, not important. Okay, so the rest of the talk will be going through like command lines and things. So if there's any questions about the algorithm, now's the time, I think. Okay. So it's a 2-part thing like everything else. You run one thing to build the model, VariantRecalibrator, and you run another thing to apply the model, which is called apply recalibration. So that's basically it. You run it for SNPs and indels independently. So we build a separate error model for SNPs and a different one for indels. And so the way that works is you just run it once, and then you run it again. So you run it once with mode equals SNP, and you run it again on the same call set with mode equals indel. And this just does SNPs and indels independently. And in the end, you have recalibrated SNPs and recalibrated indels. And what I mean by recalibrated is that these variants they're all still present in the call set. Some have been filtered out based on your the filter flag is marked based on your probability cutoff, whatever you wanted. But every variant has annotated inside its what's called the VQS LOD, which is the likelihood that it's real based on these Gaussians. So you can use that statistic in your downstream analysis if you'd like. Okay, so now we have this two-part thing, so let's go through them. So now we have variant recalibrator and apply recalibration. And before I go on, I'll just warn you that up until now, everyone has said that the command line is very simple, but this one, unfortunately, is not simple. Sorry. So don't faint or anything, but here we go. So there's lots of parameters to this thing, because it's supposed to be this extensible statistical machine learning package, right? Where you can play with machine learning via this thing. And so that's why it's quite complicated. But there's no reason to copy all this down, because we have this we have a best practices wiki page where we give the exact command line for an exome project versus a whole genome project, and you can just use that as our recommendation on what to do. So let's go through it. So we call it the variant recalibrator. Like always, we give the reference. We're going to give the input call set now. So now this is what came out of unified genotype or haplotype call or whatever. That's why it's called the raw SNPs. We're going to give these resources, which are going to be the training data sets. And I'm going to have a whole slide on the next slide to show you that. But you're going to say, use this call set as training and use this call set to decide if it's truth and all this stuff. And we'll go through that. Next, you're going to specify all the dimensions that you want to use in the clustering. So I showed you this two-dimensional plot, but it's really, that project was 8 dimensions, right? So you specify which statistics you want to use. So in this example, we have depth, quality score divided by depth, the Fisher strand bias score, and the mapping quality rank sum test score. And there's tons of these things, and you can say, I want to use this one and this one and this one, and you just type them all out. You'll say mode equals SNP for the first time, and then mode equals indel for the other time. And then the output of this thing is going to be the model, which is the recal file. So that's a file. That's going to be the input to the next step. This tranches file is how you see those tranche pictures that I showed, how you see this trade-off. So it'll show you, if you were to cut at 90% sensitivity, you would get this thing. If you were to cut at 95% sensitivity, you get this thing, and on and on and on. And so that's that file. And then we have lots of plotting things that I'll show you in a minute that also comes out. So it'll show you plots of the model. It'll show you what the distributions of the annotations look like. It does lots of cool things. And so that's that file.
Do you have to separate the DCF for SNP and indels? Because here you have raw SNP DCF.
Right. So the question is, do you have to have a separate SNP and indel call set independently? And the answer is no. So you can just give your one call set here, even if it has SNPs and indels in it. And because you're saying mode equals SNP, it's only going to use the SNPs to do its model. So you don't have to. Alternatively, you can split out your call set. You can have a SNP call set or an indel call set, and you just have so many files, and I don't like that. But if you want to do that, go ahead. But I like to have everything just as one. You feed it in, and it decides to only use the SNPs, and then next time it's going to only use the indels. That's how I like to do it, just because I don't like a lot of files. What's up?
What happens if you're missing annotations, like you have custom annotations, but they're not in other, in your files, you also bring in 1,000 files and go to the
Yeah. So these annotations, they have to be annotations that are present in your input call set. So because you're clustering the values that are found in your input call set. So It doesn't need to be in your training data set because the training data, you're just going to say, if I overlap that training data, I'm going to mark myself as training data. You're not using the values of that annotation from the training data. So that part doesn't matter. What will happen, though, is if you specify, let's say I said AN, my favorite annotation number 1. But that thing is not present in any of the call sets. What it's going to do is it's going to like look through all the calls and be like, wait a minute, you got nothing here, and it's going to tell you that. It's going to give a what's called a user error and say this value was not found in any data, so what can I do? That sort of thing. The other thing to say though is that if let's say some of your variants are missing a particular annotation, like let's say for whatever reason some of your variants are missing, what's a good one to miss? Mapping quality rank sum test. So for whatever reason there was no data there, so it couldn't calculate the statistic, so it's just missing. That's fine, because what's going to happen is the procedure is smart enough to sort of like marginalize over missing data. And so that's pretty cool. But you can't be missing everything, obviously. You could be missing some things. Make sense? Okay. Okay, so then the next thing then is to go over how you specify all this training data stuff, which is quite complicated, so here we go. And this is just an example, so don't get hung up on all these like numbers or any particular value. This is just an example. You can go to our best practices page to find the exact command lines that we recommend for different projects. But the idea is that there's 3 different kinds of things. There's known data, there's training data, and there's truth data, right? So you're going to say known equals something, training equals something, and truth equals something, right? And so the idea here is first, known data. So known data is there only as a way of calculating things like dbSNP rates or transition transversion ratio of known versus novel and things like that. It's only for display purposes. It's not used in the algorithm in any way. It's only because people like to get warm and fuzzy feelings about their call set about like, oh, I have a 90% dbSNP rate. That's like normal or whatever. So it's only there as a way of like QCing your call set to know so that you can say dbSNP is my known data and I'm using dbSNP build 137 and I want to know what is my like matching rate to that call set. That's all that's there for. Cool. The next thing is training. So the training data is very important. That's what's going to be, that's what you use as your positive labels in your Gaussian mixture model. Those are going to be the things you cluster. You're going to find all the calls in your input call set that overlap the training data, and you're going to cluster those things. So we say training equals true, training equals true, training equals true. So we're going to use HapMap, this Omni SNP chip array, and this call set from the 1000 Genomes project. We're going to treat those as all true variants, and we're going to cluster them. And that's that. Okay, the final thing is truth. So we say truth equals true, truth equals false, whatever. And the purpose of this is to say, is sort of where to decide to make this cutoff. So if I say I want a 95% like sensitivity to my truth set, you're going to say, what is, you're going to use these variants, the variants that you specify truth equals true as those calls. So you're going to say I want 95% sensitivity to this HapMap call set. So you're going to say at what level, at what probability do I achieve 95% overlap with HapMap? So that's how it works. So the first one is just for display purposes, for QCing. The second one is the actual training with the Gaussian mixture model. And the third one is for making this cutoff of deciding when to draw this line and when to start filtering variants is based on this last one. So the question is, do we always have just one truth or can we do whatever we want? And the answer is you can do whatever you want. So if you want to say I want a 95% sensitivity to both HapMap and the Omni, you would just say truth equals true and truth equals true. If you wanted 95% sensitivity to all of these things, you would just say truth equals true to all of them. Whatever you wanted to do. What's up?
So it seems like, what would be the trade-offs if you had the truth true for all those 3 that you use in training versus just one?
Yeah, so the question is, why don't we just say truth equals true for everything? If we're training on it, why not also call it true? And the thing is, clustering things together and building a Gaussian mixture model is one thing, and like you can build a probability distribution in cluster, but and things in that cluster, even things in your truth data can be off of the main distribution, and maybe it's and maybe those variants are not real, right? Even variants in your training set may not be real, but calling something truth equals true and like saying you want to use that as in deciding where to make this trade-off is much more harsh, right? So I would say that you want to be very sure the truth equals true is a very, very, very high quality call set, whereas the training data can be a little less high quality. Does that make sense, what I'm trying to say?
Yes, it makes sense. So for these resources, I mean, is there some sort of a description that would actually inform us how true they are?
Well, so that's sort of, yeah, so the idea is how do we know which call sets to trust and all this stuff.
It seems like you, with the half map, is a high quality, but is there any comparison, like the Omni resource, is it still okay, or
Yeah.
what's the quality of the Omni?
Yeah. So I mean, for this, you would have to just go to the publications where these things were published and decide for yourself what quality you think they are. So the other thing we haven't talked about is this prior, and these are sort of my estimates of how true they are. So I would say that the hat map is much higher quality than Omni, which is much higher quality than Thousands of Genomes thing, and then dbSNP is quite low quality. And so it's sort of, unfortunately, it's sort of up to everyone on their own to decide how much they want to trust each call set. Sorry, I don't have a better answer, but I'm trying to be like a skeptical scientist, though. Like, you sort of have to decide for yourself, I think, how much you trust these call sets. But anyway, this is the order that I put them in, is in terms of their truth rate.
So when you mentioned the 95% sensitivity cutoff, is that number solid or is?
Well, so the question is about this 95% true sensitivity cutoff. And so there's definitely a default value, which I think is 95%. But you could decide in the command line. You'll say, I want a true sensitivity of X, right? And we have recommendations for that as well on the website. I think on the website, actually, we're recommending 99% sensitivity because we've gotten much, much better over the years at actually doing this stuff. And so you can actually achieve, depending on your project, of course, you could actually achieve 99% true sensitivity to a call set like HapMap and still have a very low false positive rate. Of course, if your project is like dealing with rare variation in some population that's not in 1,000 genomes or HapMap, like whatever, Papua New Guinea or something, then maybe you can achieve 99% sensitivity. You just have to, these are things that you have to sort of decide on your own, I think. Does that make sense, the trade-off? Okay, so after we've built the model using our input VCF file and all these training sets, now we have the recalibration file, the tranches file, and the plots, and now we're going to apply recalibration. So we're going to take our input file, we're going to use the model, we're going to now annotate every variant with its probability score, and then we're going to filter variants based on whatever true sensitivity you wanted to specify. So, and that looks like this. So you give the input call set, you're going to say mode equals snip again. You give the recal file and the tranches file. That was the output from the previous step. Your output is going to be the new VCF file that's now annotated with all this fancy information. And then you're going to say what true sensitivity level you want to filter at. Maybe the default is actually 99. I forget, but whatever it is, that's what you put there. What's up? Yeah. It does, yeah. So what you'll see is that every variant, oh, it says it right here, actually. Every variant is annotated with this LOD score. So in its info field, you'll see VQS LOD equals something, -1, 0.1, or it'll say like whatever it is, right? And so that's just a score. and that you can use however you want to use it. You can decide for yourself where to filter. You can maybe plot that distribution and see what you like about it. People have started, which is really cool, people started using that statistic in their like association studies, which is pretty awesome. So you can do whatever you want with it. Make sense? Okay. What else is there to say? We have a pro tip. Wonder what that is. Oh, so the idea is that you could just run it once with SNPs and then again with indels. So you don't have to like separate your call set, like I said before. You can just use the one call set, run it with SNPs, run it with indels, and then you're done. Okay. And so then the question is, how do we know if it worked, right? Because this is like, you just feed it this thing, it spits out a bunch of numbers, and how do you know if anything happened? So the first thing you can do is check to make sure that filters have been applied to your VCF file. So what you'll see is that some variants have been filtered out, because not every variant was real, and so in order to achieve a 99% true sensitivity, you had to filter some stuff out, right? And so you'll see filter fields now in your VCF. In addition, you'll see that every variant has a VQS LOD attached to it. And then the final thing is that as a part of the procedure, the VQSR is going to output all these plots, all these mixture model plots, and you can look at those to look at like the distribution of the data and stuff. And this is what it looks like. It's pretty cool. So here I'm plotting the mapping quality rank sum test against the read position rank sum test. And you have the heat map, which shows you the probability density function for the model that was learned. You have these were the dots are now the actual variants, and it shows you which variants were retained in your call set and which ones were filtered out. And first of all, you can see that it's kind of beautifully Gaussianly distributed. It looks pretty good. And the things that are way off in the distributions are the things that were filtered out. It's showing you the knowns and novels. So most of the knowns were looking pretty good, and most of the novels are most of the things that were filtered out were novel. And there are some known sites that were also filtered out, things like that. So you can spend however long you want looking at this stuff and trying to decide if it looks, like it did a reasonable job of deciding, which things were real and which things were false, right? Like if your distribution were just a bunch of random point clouds and there were red and black dots everywhere, I would say it probably didn't work so well. Make sense? All right. And so now we've completed variant recalibration, and I'll take some questions if anyone has any. What's up?
You have an organism where you don't have known SNPs.
Yeah, it's pretty cool. So there's what's really been fun about this thing is that I started by saying you needed all this truth data, right? You're like assuming that like 1,000 genomes is real and everything that matches 1,000 genomes is good, that sort of thing. But it actually works pretty well even with no training data at all. Because the idea is that variants that cluster together are going to be real, and the things that don't cluster, the things that are way off, are going to be false. And so even with no training data whatsoever, it can do a pretty decent job. I'm not claiming that you're going to get fantastic results. You're certainly going to get better results if you have actual training data. But it still works pretty well, because the core concept is that things that look like each other are probably real. So anyway, there's lots of things you can do. One thing that, like the fungal groups at the BroadE, I don't know the proper name, I'm sorry. But one thing that they've been doing is they'll take, they'll call variants with like SAM tools and GATK and whatever, all these callers, and sort of like intersect the results and say that the things that were called by all these callers are probably real, and use that as truth data. And it works pretty well.
It's truth or it's training?
Oh, sorry, as training data and as truth data. So you would say, my intersection call set, training equals true, truth equals true. It has a prior of like Q6 because you don't really trust it. And it actually works pretty well to do that sort of thing. Because the idea is that the core concept is that things that were called by all these callers are probably real because they were seen by all these callers. And so that works pretty well. Any other questions?
Well, the cancer people have a similar sort of tool that they use since they don't output the dataset file.
So the question is, what do the cancer people do? And unfortunately, I don't know quite so much, but I think the basic idea is that they use annotations like this, like strand bias and QD type things and all this stuff. But I think they have like, I think they like have these thresholds for like what they decide is good and not. I'm not sure. Yeah. I think it's basically hard filtering. Although it's probably more fancy than that. I'm just not sure what it is. What's up?
So I guess the one question I already asked about on the RNA-seq, recommending similar to the cancer data, if you have low frequency allele source, you're just looking at genotypes. It's either homozygous or heterozygous. If you took out the filter for I guess how to look at the lead depth.
Yeah.
Would it perform better for that type of data?
I think so. So, like I said, some of the annotations are certainly used or not used depending on the data type. So, I think probably the most analogous thing is going to be exome capture. So, in exome capture, we don't use depth because in that sort of process, depth varies just naturally. And so, it's not indicative of error. Having an excessive depth or having a low depth is not indicative of error. The same way it is in whole genome sequencing. And so, so there are certainly things like that you can just take out of the model, and it'll still use things like strand bias. Strand bias is probably indicative of error, right? Or even you could do strand bias and like this read position rank sum test, and just use those two statistics, and you'll probably still do a better job than just like hard filtering on your own, right? You don't have to be quite as fancy as we're being for the whole genome process, but you can still use this procedure and to sort of experiment and decide for yourself what works the best. I think it'd be really fun, actually, to make this thing work with RNA-seq data and cancer data and all this other stuff. It would just take someone who's willing to experiment with these different annotations and et cetera.
Just mentioned that you need enough data.
So I was reminded that this is a statistical machine learning process, and you sort of do need a whole call set's worth of data in order to make it work, right? If you only have, like, let's say, a targeted sequencing experiment where you target a gene and you made a call set and you have like 100 variants, you're probably not going to be able to do much clustering with just 100 dots on the point cloud, right? So what I'm showing you here is like thousands or tens of thousands of variants in order to cluster and to make those distributions. So keep that in mind too. If you have 100 variants, you're not going to be able to do much clustering.
And this is the reason that if you have the set exome intervals, we recommend to add lots of genome samples, so you will have enough points, enough things to do the clusters. And what do you do if you don't? Then just go to hard filters. So people, if you don't have enough data to do that, then your only option is to go back to hard filters.
Anything else? Well, thank you very much for your attention.